Hello, everyone. Welcome to week 3, lecture 5. And today's topic would be methods of identification, part 1. So learning objectives for this particular topic would be, at the end of the session, the students are expected to be able to discuss concepts and principles of the techniques by which bacteria are identified in the laboratory. So this is part 1, and we will be covering microscopic until culture methods. Now, to provide you with an idea of the entire lineup of methods that we usually use to identify organisms in a specimen. Okay? So these are the methodologies. Okay? So we start out with microscopy and staining. So this method allows us to observe the morphology, viability, and motility of microorganisms. Okay? So usually it would provide us with the general groupings as to whether they are spherical in shape, whether they are gram-negative or gram-positive, whether they are bacilli or spirals. Okay, now this is usually followed by culture methods. Culture methods would allow us to see the macroscopic characteristics of the organisms, the type of colonies that they produce, the different environments that they would need for survival, the different incubation methods, temperature, and environment that would be useful for an organism in order for them to grow in an artificial media or artificial environment. Okay? The culture results may provide us with the genera or the genus of an organism, but we have to move further so that we will be able to identify species of the organisms. So this can be acquired through biochemical tests. Okay? So biochemical test demonstrates the enzyme system in each organism. Okay? There are specific sets of biochemical tests for a particular group of organisms. Okay? Then this may be followed by serological test, which is done to identify bacterial serotypes. So if the results of the first 3 methods would lead us to an organism with several serotypes, so it would require serological testing. Currently, we have molecular methods. Okay? So these are usually involves DNA sequencing techniques. These brought about newer methods to identify microbes more precisely. Okay? And the good thing about it is that it would not require us to culture microbes. Okay? So therefore, it's quite useful for non-culturable organisms. Okay? Also, molecular testing is great for identifying fungal organisms and viruses. Okay? So it actually provides a more robust tool set in identifying infectious agents. Animal inoculation is also included. However, we do not usually use this in the clinical laboratory. Okay? Most of the time, animal inoculation is quite useful for research laboratories. So it's done to investigate the effects of the inoculated material. So first stop, microscopic method. Okay, so microscopic methods involve smear preparation. So smear, by definition, is a thin film of microbial solution on a glass slide. Okay, so this would allow us to view organisms or microbes in different states. Okay, we can view them in their living state. Okay, this is quite useful in visualizing their size, their shape, their arrangement, and of course, their motility. Okay, so smear preparation that would allow us to view organisms in their living state includes wet mount preparation and the hanging drop method or preparation. Okay, so allow me to show you a video of how wet method or wet mounts are prepared.
This demonstration is how to prepare a wet mount. The purpose of doing a wet mount is to check for the motility of a cell and also the size of a cell. Make sure your gloves and your goggles are on. I'll be demonstrating how to do a wet mount when the starting material is a liquid, and then when the starting material is coming from a colony on a Petri plate. Start by taking a slide from the box. You will need a cover slip. Cover slips are squares of either glass or plastic, and they are separated by pieces of tissue paper. When the starting material is a liquid, take a drop of the material, put it in the center of the slide. Take your cover slip, hold it at a 45-degree angle, touching the slide, and gently drop it. When the starting material is a colony on a Petri plate, you will take a slide, and you're going to put a drop of deionized water in the center of the slide. Then, using aseptic technique, you'll be using your inoculating loop. You'll need to sterilize your inoculating loop by putting the loop into the flame of the Bunsen burner. Remove the lid from the plate. If there's an area on the plate where you can touch the loop to cool it beforehand, that would be a good idea. Touch the surface of a colony. Immediately put the lid back on the plate. Put the loop in the drop of water. Spread it around about the size of a dime. Sterilize the loop before you put it on the bench and turn off the Bunsen burner. Take a cover slip, hold it at a 45-degree angle, and gently drop it on the sample.
Okay, so that's how simple wet mounts are prepared. Okay, so if it comes from a liquid media, no need to add the ionized water or sterile distilled water. Okay, so we only use the ionized water or sterile distilled water if our samples would come from a plated media. Okay, now let's take a look at what we would be looking at if we have had prepared wet mounts in the laboratory. Do you see that? Okay, there. You can actually see rods. Okay, there. That's great. Okay, so you can see a long rod-shaped bacteria. Okay, that's running. You can see one that's tumbling. Okay, so remember the run and tumble movement of a flagellar organism. Okay, so when they need to change direction or when chemotactic substances are found in a different direction, they tumble and then move towards that direction if it's an attractant, and away from that direction if it's something to get away from, like antibiotics. Okay, so technically that would be what this video contained until the end. So we'll stop it right there. Okay, so that's how microorganisms, moving microorganisms, would look like when prepared in the fresh or living state. Okay, so this is another preparation of microorganisms that would demonstrate their living state. Okay, so this is the hanging drop preparation. Okay. So in this video, they'd be showing different types of slides. So you have they would show you the actual hanging drop slide. And then if you don't have the hanging drop slide, they did show samples where you can actually create your own. hanging drop slide. So this is the actual hanging drop slide. This one is made out of adhesive, and then this one is made from paraffin wax, just to create a hollow. So cover slips are prepared, and a loop full of the organism or specimen would be placed on the cover slip. Of course, practice aseptic technique. Never lay down your inoculating loops without sterilizing them. Okay, so paraffin wax is added to seal, so it would serve as a sealant for the cover slip, the glass slide. Okay, so not too much, just enough to ensure that the droplet would remain stuck to the cover slip. And then the slide is inverted. Allow the cover slip to stick to it. Okay, so the drop of organism actually hangs on the cover slip because of the presence of the paraffin wax. If you don't have paraffin wax, you can actually use petroleum jelly. Okay, so for initial viewing, you'd have to look for the edge of the droplet. Okay, so it's easy actually to look for the edge of the droplet. Okay. So we need to look at the edge of the droplet because that's where the organisms would be there. Okay. So the organisms are actually stuck on the edge of the droplet. So some are moving away from the edge. Okay. Okay, so you may also prepare organisms and view them in their fixed state. So preparing fixed smears mean that organisms are no longer viable because you'll be subjecting them to either heat or chemical in order for them to become fixed. Okay, so they're no longer viable, but they can be now stained, and so we can view them more clearly. Okay, so to fix smears on slides, we could apply heat fixation. Okay, so generally this preserves the overall morphology, but not the internal structures. Okay, so heat denatures the anything that's protein. Okay, so like the proteolytic enzymes within the cells. Okay, so when the proteolytic enzymes are denatured, this would lead to inhibition of autolysis. Okay, so in the process, you'd be able to preserve the organism. Okay, then you can also make use of methanol fixation. So this one would prevent lysis of RBCs. It also is useful in avoiding damage to all host cells, and it results in a cleaner background, okay, compared to when we do heat fixation. Okay, so it's strongly recommended for all clinical material, all samples, especially for urine, because we will be making use of the sediments, and there's a tendency for the sediments to slide off, okay, or be removed from the smears in the process of staining. Okay, so to avoid that, you can actually make use of methanol. But the sad thing is that methanol is quite toxic, and they're not really recommended to be used in routine testing. Okay, but the purpose of using either heat or methanol fixation is to kill the organisms and preserve them at the same time. Okay? Also, fixing smears on the slides allow them to anchor more effectively on the glass slide so that even if they are washed several times in the process of staining, they'd be more efficiently stuck to the glass slide. Okay? So once you have heat fixed or methanol fixed your slides, okay, you can now proceed to the next step, which is staining. So staining is the process of adding stains or dyes for enhanced visualization of the microbes. It's more interesting to view organisms with color. Okay? The smallest they are, okay, the presence of stains or dyes or colors would help in finding them more efficiently on the smears. Okay, so microbial staining methods can be divided into two types, as was discussed in the laboratory. Okay, so direct methods are techniques which allow the stain to come in contact with the organism to be identified or object to be viewed. So we directly color the organism and leave the background colorless. The other type would be indirect staining, wherein the background is the one that's stained, leaving the organism to be identified as colorless. But in effect, it is actually helpful because the light or colorless organism appears highlighted against the dark background in the indirect staining. Okay, so under direct staining, we have simple staining methods, which would employ the use of only one color or one stain, like crystal violet or methylene blue, okay, to able to achieve the colored appearance of a fixed smear. Okay? However, it's not widely used in the laboratory. Okay? Special staining methods can be either direct or indirect. Okay, so one example of a special staining method that's indirect is your India ink. Okay, so it's a special staining process for the identification of capsules. Okay, so that's indirect. However, the rest are actually direct staining methods. Okay, so special staining methods are used to highlight or showcase or demonstrate the presence of special structures that cannot be ordinarily seen or regularly seen in gram stained slides. Okay, so examples of this would be the flagella, um, the capsules, the spores, and the granules, no metachromatic granules. Okay. However, they're not commonly performed also in the lab. Okay, so in routine laboratories, very rarely or very seldom are special staining methods requested or performed. Okay. Now, differential staining method is the most common staining technique employed in the microbiology laboratory. This includes gram staining and acid-fast staining methods. So in this staining methods, we employ the use of several chemicals, several stains to show contrast and differentiation of organisms that may be found in a single smear. Okay. So moving on. Okay. These are examples of special staining methods. Okay, for the capsule, you have Welch, Anthony's, Gins, His, Tyler's, Muir's, India ink. For cell wall, you have Dyer stains. For metachromatic granules, you can use Albert's, Lubinsky, Lamb, Neisser's. Flagella, you can use Leifson's, Fisher-Kahn, and Gray's. And for endospore, you can use Dorner's, Schaefer-Fulton, Wirtz-Conklin, and the glacial acetic acid. Okay, so this is how special stains would look like. Okay, so we have the negative staining method to demonstrate the capsules. Okay, so it's really good actually. So you can see that the background is fully colored, while the organism or the structure to be viewed is left colorless. Okay. So it's colorless against a colored background. Okay, then this is a flagellum. Yeah? So flagellar staining to demonstrate that this is an amphitrchous organism. So the tuft of flagella on both ends are well demonstrated. And then we have the endospore staining method. So the blue dots that you see in this picture are actually the endospores. Okay, so they are highlighted against a pink background. So moving on to differential staining. Okay, so again, as mentioned earlier, differential staining is actually the most widely used staining technique in the microbiology laboratory, and it includes gram staining. Okay, so gram staining is based on the thickness and chemical composition of the cell wall. So through the staining methods, it can differentiate bacterial agents into two general groups, the gram-positive organisms and the gram-negative organisms. So it was discussed previously in lecture 1 on how the thickness and the chemical composition would determine the gram staining reaction. So as previously mentioned as well, that this staining method was developed by a Danish bacteriologist, Hans Christian Gram, who devised a test to identify whether or not a bacteria had a peptide glycan wall. So in his test, a dye was introduced to the bacteria. So he did a staining process. If the bacteria had a peptidoglycan cell wall, it absorbed the dye and turned purple. So it tested positive for peptidoglycan. If it did not turn purple, it tested negative for peptidoglycan, meaning its peptidoglycan layer was thin or there wasn't any peptidoglycan layer at all. Okay? So as this method was adopted, okay, the resulting categories were called gram positive and gram negative. So this method of gram staining is still widely used. It's a standard procedure in microbiology. Okay. So the chemicals that we make use of in routine gram staining reactions, gram staining process are the following. Okay, so we make use of the crystal violet as the primary stain. So it stains both gram positive and gram negative cells with purple. Okay, because the dye enters the cell wall of both cell types. Now, when iodine, Gram's iodine, which serves as the mordant, is applied onto the smear, it forms large crystals with the dye. Okay? So these large crystals will be too large to escape out, right back out through the cell wall. Okay? So it gets trapped and locked, and it will be locked together with crystal violet and teichoic acid and magnesium RNA. So all in all, they'd make good partners. So they'll get stuck onto the peptidoglycan layer of the gram-positive organism. Okay? So the application of acetone alcohol dehydrates the peptidoglycan layer of gram-positive cells, making the pores close, and therefore the crystal violet iodine complex becomes stuck or trapped in the peptidoglycan layer. So it becomes, the complex will make the cell wall impermeable. Okay? So it restricts the absorbance of other materials or chemicals into the bacterial cell wall. Okay? Then for gram-negative organism, okay, the effect of acetone alcohol is quite negative towards the lipopolysaccharide layer. So the acetone alcohol would actually create pores on the lipopolysaccharide layer, allowing the acetone alcohol to enter into the thin peptidoglycan layer, removing the crystal violet in it. Okay? So leaving or rendering the cell wall of the gram-negative organisms colorless, so that when we apply the counter stain, okay, the colorless gram-negative cell walls will absorb the color of the counter stain, which is red. Therefore, gram-positive are stained purple and gram-negative organisms are stained red. Okay, so this is how a gram stain smear would look like. So it would show purple rods if you have gram-positive rods. Okay, so purple rods. We also have a cocoa bacilli here. We have probably these are diplophoci, okay, or short chains. Okay. So they're also gram-positive, purple. And then you have vibrios, okay, curved rods that are red. Okay. So gram-negative vibrio. So these are the gram reactions, of organisms that are stained with gram staining. Now, gram staining has undergone modifications, okay, over time. So initially, what Hans Christian Gram used was methyl violet as the primary stain and acetone as the decolorizer and dilute carbolfixin as the counter stain. Okay, so there were two identified modifications. We have the Hawker's modification, which changed, or made use of crystal violet as primary stain instead of methyl violet. Okay, for the mordant, Brahms iodine was used, and then for the decolorizer, a combination of acetone alcohol was used. Okay, so and the counter stain was also changed into safranin. Originally, it was dilute carbolfixin. Now, the Burke's modification, okay, retained crystal violet as the primary stain and safranin as the counter stain, and Brahms iodine as the mordant. However, it changed the decolorizer into sodium bicarbonate and ether acetone. Okay, so that's Burke's modification. Okay, so general rule. Okay, everyone should memorize this by heart. Okay. So the general rule for gram staining are as follows. All cocci are gram-positive except Neisseria, Branhamella, Moraxella, and Veillonella. All bacilli are gram-negative except Mycobacterium, Corynebacterium, Lactobacillus, Listeria, Clostridium, Bacillus, Erysipellatrix, and Nocardia. All spiral bacteria are gram-negative when stained. Okay. Now, there are several theories trying to explain why those were the gram staining reactions of different organisms. Okay, so why are gram positive purple? Why are gram negative red? So we have here 1, 2, 3, 4 theories. So let's go with the first theory, which is the magnesium ribonucleate theory. So it is said that a compound of magnesium ribonucleate and a basic protein concentrated at the cell wall helps gram-positive bacteria to retain the primary dye. Okay. However, this particular material is not found in the cell walls of, or cell membrane rather, of gram-negative organism. So if magnesium RNA is present, it binds well with the crystal violet iodine complex, creating an insoluble compound. Then we have the Binyan theory. So this one, according to this one, cell walls of gram-positive organisms are less permeable. Okay, so some substances are not easily absorbed into the thick, compact peptidoglycan layer, especially when another substance is trapped in it, like the crystal violet. However, the lipopolysaccharide layer of the gram-negative organisms, okay, their cell walls, the cell walls of gram-negative organisms, are sensitive to acetone alcohol, so rendering the cell wall to be permeable. So remember, when when the acetone alcohol rather is applied onto the stained cell walls of gram-negative organisms, holes would be created by the acetone as a reaction to the presence, as it actually lipopolysaccharide itself will actually react to the chemical acetone alcohol. So pores will be produced on it. And because there are pores, it would make it easier for the acetone alcohol to penetrate deeply, into the thin peptidoglycan layer. Okay? Then we have the Stern and Stern theory. Okay, so for the Stern and Stern theory, this is something to do with the isoelectric points and pH. Okay, so for this theory, gram-positive organisms are said to have a low isoelectric point, making them acidic. And when a material is acidic, it binds perfectly well with basic dyes like crystal violet. Gram-negative cell walls are said to have increased isoelectric points, making them basic. Okay? And basic dyes, basic materials does not bind well with basic dyes, okay, like crystal violet. Okay? Then we have the lipid theory. Okay? So the lipid theory believes that in the cell wall of gram-negative bacteria, okay, there's the lipopolysaccharide or the outer membrane, okay, which has, of course, lipid. Okay, but it does not have teichoic acid. Okay? So lipids are easily penetrated by the action of acetone alcohol, okay? Leaving the um, allowing materials to come in and come out of the cell walls. Okay, now unlike cell walls of gram-positive organisms, they don't have that much lipid content, but they do have teichoic acid, and this teichoic acid binds perfectly well with the crystal violet iodine complex, okay, allowing the binding or allowing the primary stain to get stuck, no, to the peptidoglycan layer. So again, it might not have that much lipid, but it does have a high teichoic content, which works well with crystal violet iodine complex. In contrast, gram-negative cell walls does have high lipid contents, but it's easily affected by acetone alcohol. And there's nothing for crystal violet iodine complex to latch onto apart from the thin peptidoglycan layer, but there's no teichoic acid. Okay, so apart from the thin peptidoglycan layer, okay, there's nothing to hold on to. There's no magnesium RNA, there's no teichoic acid, so nothing to bind a crystal violet iodine complex well onto the peptidoglycan layer. Okay? And also, there's only a really, really thin layer of peptidoglycan, so that when acetone alcohol is applied, it perforates the lipopolysaccharide layer, enters the thin peptidoglycan layer, and removes the crystal violet iodine complex. Okay, so that explains these series try to explain why gram-negative organisms are red and gram-positive organisms are purple. Again, magnesium RNA theory maintains that the mgRNA binds well with crystal violet iodine complex, therefore it's not easily removed when decolorized. In the same manner, in Binyan theory, once a material penetrates or is absorbed into the peptidoglycan layer, it cannot be easily removed because cell walls of gram-positive are believed to be less permeable. For the Stern theory, due to the low isoelectric points of the gram-positive cell wall, it binds perfectly well with the crystal violet, hence it retains the violet color. Now, for the lipid content, because it has a very high teichoic acid level, okay, the low lipid content is not a problem because it's the teichoic acid that binds well with the crystal violet iodine complex, hence allowing the crystal violet to retain in the cell walls of the gram-positive organism. Okay. For gram-negative, they don't have mgRNA, so nothing will bind to the crystal violet iodine complex. Okay. Their cell walls are permeable, so even if they have thick lipid layers, no? Lipid polysaccharide, lipopolysaccharide layer. It's sensitive to acetone alcohol, so it gets perforated and it allows materials to come in and out of the cell walls easily. That's why the purple color is removed and is replaced by the red color, taking of the safranin. Okay. Also, the high isoelectric points of the gram-negative cell walls allow them to makes them basic, you know, and is basically responsible for not being able to attach to crystal violet iodine complex. Okay. And then finally, because they don't have teichoic acid, okay, even if they do have very thick lipid layers, okay, it's useless when acetone alcohol is being applied. Okay. So teichoic acid is not seen or is absent in the cell wall. So there's nothing for the crystal violet or iodine complex to attach to, so that it would be easily removed during decolorization. Hence, gram-negative organisms take up the color of the secondary stain. Okay, now there are certain reasons why there are errors, no, in performing gram staining. So there are a lot of the errors actually are when gram-positive organisms become accidentally gram-negative organisms. So one of these is when we use acidic grams iodine. Okay, so acidic grams iodine would destroy the peptidoglycan layers. Okay? So there will be no crystal violet iodine complex because it's too acidic. It damages the peptidoglycan layer. So crystal violet will not have anything to latch onto and it will not be retained in the cell walls. Also, if there's insufficient iodine exposure, okay, it would also lead to removal of the crystal violet. Okay, so the amount of the mordant available is important for the formation of the crystal violet iodine complex. So the concentration of the grams iodine has to be just right. Okay, so that it won't get easily decolorized, because the lower the concentration will be, the easier for the crystal violet to be removed, you know, from the cell walls. Then when you are when you have gram-stained old, you know, bacteria or dying bacteria, or those who have undergone autolysis. So here, their cell walls are no longer intact, no? And their peptidoglycan layer might have been damaged or destroyed in the process. Okay, so therefore, your crystal violet will have nothing to latch onto. Okay, so once it enters the cell wall. Overheating can also result to gram-positive becoming gram-negative. Okay, so during heat fixation, remember not to do it excessively. That's why we have to simply let the slides pass along the flame from time to time, moving back and forth. We're not supposed to leave our slides, no, on top of the flame. It will break our slides, but in the process, you know, it will also destroy, okay, the cell morphology. Okay? So it will damage the cell wall and the peptidoglycan layer along with it. Okay, and then low concentrations of crystal violet. Okay, so crystal violet up to 2% concentration can be used successfully. Okay, so standard solution is around 0.3%, okay, which is good, provided that you don't decolorize more than 10 seconds. Okay? But it would be best to use something higher than 0.3%. So you can actually use up to 2%, okay, crystal violet. Okay, then what else? A removal of magnesium RNA with precipitation from bile salts in culture media. So bile salts in culture media are also bad for the magnesium RNA in cell walls of gram-positive organisms. So when this is removed, remember, it binds well with the crystal violet iodine complex. So when it is removed, okay, or has been precipitated by bile salts. Okay? So there will be a lack of something for your crystal violet and iodine complex to latch onto in the cell walls. Okay. Then over decolorization, okay, so prolonged decolorization would also lead to gram-positive organisms becoming gram-negative. Okay, so if you're not yet skilled enough, okay, or you're not yet familiar, okay, with the decolorization process, you may use 95% ethanol, okay, to decolorize. Okay. Um, because it decolorizes more slowly. Okay? So those who are used to the process of decolorization, such as those that are experienced in the laboratory, they can freely use acetone alcohol. Okay? So you need to practice and practice, you know, so that you'll be able to gauge properly, yeah? How long do you need to decolorize your smears. Okay, and then we have excessive washing. Okay, so remember in the course of staining, different chemicals are applied, and in between, you need to wash your slides with water. Okay, so the crystal violet stain is susceptible also to washing out with water. Okay, so remember, do not use more than a 5-second water rinse rather at any stage of the procedure. Okay. And then, of course, excessive counter staining. So as a counter stain is also a basic dye. So safranin is also a basic dye. It is possible to replace the crystal violet iodine complex in the gram-positive cell walls. Okay, so especially when we overexpose it to safranin. So remember, it's either from 30 seconds to 1 minute, no? 30 seconds to 60 seconds is the usual contact time of safranin. Okay? Sometimes you tend to forget that you were staining, so you left it there for 2 minutes. That would also lead to replacement of the crystal violet iodine complex with safranin. Okay? Now, at times, you can also identify, you know, errors involving gram-negative organism becoming gram-positive. Okay, so inadequate decolorization is the most common reason for this. Okay, so sometimes, if you have experienced having your smears over decolorized, you'd be traumatized to perform decolorization, ending in an inadequately decolorized smears. Okay, so don't be afraid. You can prepare actually several smears, okay, of your specimen. Okay, so if you're still practicing, you can prepare several smears and check. You know? So follow the decolorization time rather. Okay, and sometimes also when thick smears are prepared. Okay, so even if the organism is gram-negative, you will not be able to remove, you know, the excess crystal violet that has stuck onto a really thick smear. So you'll end up with a gram-negative organism that has become gram-positive simply because, okay, even if you have decolorized it for 1 minute, the excess crystal violet is still stuck because the smear is too thick. Okay. Now, there are ways by which you can check the true gram staining reaction. So we have here 2 methods that are actually able to identify gram-negative organisms. So we have the LANA or the LANA test. Okay, so to perform this test, the reagent used is L-alanine. Okay, so L-alanine is used to make a suspension with the bacteria. So in a small tube, get a 1 ml of the L-alanine, add a loopful of the organism, and observe. Okay? If the solution or the mixture turns yellow, the organism is gram-negative. How so? Okay? Gram-negative organisms usually release their aminopeptidase enzyme in the presence of L-alanine. They'd be releasing 4-nitroaniline, which produces the yellow color. Okay, so if the color of the suspension turns yellow, the organism is gram-negative. Okay, now 3% potassium string test, potassium hydroxide string test is also used to identify, gram-negative organisms. So to perform the method or the process, we simply use a drop of 3% potassium hydroxide and put it on a glass slide. Now, afterwards, add a loopful of cells, okay, from a single colony, and then mix it with your 3% potassium hydroxide. Now, if the mixture becomes viscous within 60 seconds, it means that it is KOH positive. Okay? The colony is considered gram negative. Okay? So if a string-like material is developed after 1 minute of mixing, it means that the organisms are gram negative. How so? Well, this reaction actually depends on the lysis of gram negative cell in the dilute alkali solution, which is KOH. Okay? So when they are exposed to KOH, cellular DNA is released, causing the suspension to become viscous. Okay, so L-alanine, if the solution turns yellow, that's gram negative. So in KOH, if there's the formation of a string-like material, then the organism is gram negative. Okay, moving on to acid-fast staining. So this is the second most common staining method, differential staining method in the bacteriology laboratory. Okay, so there are some bacteria that have the ability to retain the primary stain and resist acid decolorization process. Thus, they're called as acid-fast. Okay? And their acid fastness is said to be due to the presence of mycolic acid in their cell walls. Okay? Mycolic acid are long carbon atom chains. Okay? So as a general rule, all organisms are non-acid fast. No? So there are only a few acid-fast organisms, and this includes, number one, the Mycobacterium organisms. So these are organisms with the longest chain of mycolic acid. That's why they are strongly acid-fast. Okay, then we have Nocardia. Okay, so Nocardia have short cell wall bound mycolic acid. Okay. Then the shortest, no? The one with the shortest chain is Corynebacterium. Sometimes, even if we perform gram staining, Corynebacterium won't be able to demonstrate, acid fastness because their chains are really short. Okay, so the process is actually developed by Paul Ehrlich in 1882 and was later developed by or improved by Zill-Nielsen method. By Zill-Nielsen rather. So Paul Ehrlich created it and improvised by Zill and Nielsen. Okay, so the chemicals that are used in acid-fast staining are the following. Okay, so carbolfuchsin is used as the primary stain. Okay. So if the cell walls have mycolic acid, carbolfuchsin binds well with this material. Okay. So in the first exposure, both acid-fast bacilli and non-acid-fast bacilli are stained red, taking up the color of carbolfuchsin. Now, since the cell walls of these organisms are rigid and waxy, it's quite difficult for the carbolfuchsin to penetrate. Okay? Now, to allow penetration of carbolfuchsin, we need to facilitate staining by adding a mordant. So in the process, the mordant here is heating. Okay? So we heat to allow the pores of the microbial cell wall to open up to allow the penetration of carbolfuchsin. Okay. Now the acid alcohol here serves as the decolorizer. Okay, so the decolorizer will remove carbolfuchsin from cell walls without mycolic acid. Okay, with those cell walls with mycolic acid, they will retain the color of the primary stain, red. Non-acid fast, since they don't have mycolic acid, will be rendered colorless during decolorization. Okay, so this is followed by methylene blue, okay, which is the counter stain. So in the presence of acid-fast bacilli, okay, the background is blue, but acid-fast bacilli will retain the red color. Okay? Now, non-acid-fast organisms are stained blue along with the matrix or the environment. Okay? So, acid-fast, red, non-acid-fast, blue. Okay? So this technique is called as the Kinyon method, the Ziehl-Neelsen method. Okay, so this is the microscopic picture of Mycobacterium leprae. Okay, so this is a single field. Okay, so the patient is actually heavily infected with Mycobacterium leprae. If this is the amount of acid-fast bacilli that we get to see per field, no, per microscopic field, then that's really, really a lot. Okay, so Mycobacterium are acid-fast bacilli. They stain red, but the environment is stained blue. Okay. Now remember, since the cell walls of acid-fast bacilli are really tough and waxy, okay, so we need to employ different methods to facilitate the entry of carbamazepine into the bacterial cell wall. So what are we going to do? We can use steaming or heating. Okay, so I will show you that process later. Okay, so heating or steaming will open up the pores of the microbial cell wall, allowing the entrance of peptide of the carbamazepine into the cell wall. Okay, carbamazepine into the cell wall. Okay, you can also, if you don't want to use steam or heating, you can remove this, okay, but do increase the concentration of phenol and basic fixin. Okay, so when the concentration is higher, okay, there's a greater chance that more carbamazepine will be able to enter into the cell wall. Then we have prolonged contact time. So you can also prolong contact time. You may not use steam, you may not increase the concentration of phenol, but instead of applying the 1 minute rule for most stains, you can increase from 3 to 5 minutes, no, of contact time. So ensuring that carbamazepine is being transported into the cell walls of the organism. Okay, then you can also add wetting agents. So when you have added wetting agents like turgitol, there's no need for heating. Okay, so wetting agents are also called surfactants. No? So surfactants are chemical substances that increases the spreading of and penetrating properties of a liquid. Okay, so this is done by lowering its surface tension. So when turgitol is added into carbamazepine, the surface tension of carbamazepine is lowered, okay, allowing it to easily flow into the cell walls of acid-fast organism. Okay. Okay. The types of acid-fast staining. Okay, so there are 4 types of acid-fast staining, but the most widely used is the Ziehl-Neelsen method, followed by the Kinyon method. So in the Ziehl-Neelsen method, heat is used as the mordant. So it's called the hot stain acid-fast method. Okay, here it makes use of carbamazepine as the primary stain and methylene blue as the counter stain. So acid-fast organisms are stained red, non-acid-fast organisms and the background are stained blue. Okay. The Kinyan method is similar to the Ziehl-Neelsen method, except that it does not make use of heating. Okay, so it's called the cold stain process of acid-fast staining. So instead of using heat as the mordant, it makes use of tergitol or wetting agents to allow the or to facilitate the absorption or penetration of the primary stain into the microbial cell wall. Okay, so either malachite green or methylene blue is used as the counter stain. Carbylfixin is still used as the primary stain. So acid-fast organisms are stained red, non-acid fast organisms and the background may be stained green for malachite green and blue for methylene blue. Okay, so allow me to show you a video of the Ziehl-Neelsen staining technique so you'll see how stain is applied in the process. So usually for acid-fast staining like Ziehl-Neelsen, we flood the smears with the stains that are used. So we use heat there, so it's placed under the smear. We usually apply it until vapor start rising from the stain. Okay. So it's washed off carefully. There, so see, the stain is visible on the smears. So decolorization with acid alcohol. So again, this is a crucial portion of the staining process. So careful with decolorization. Remember to rinse carefully. Again, flood with the stain. After 60 seconds, you're now done with the technique. So it's really easy. Sometimes artifacts from carbylfixin may stick on the smears. So you may use filter papers. You can put filter papers on top of the smears and then you flood that with carbylfixin. So those are your acid-fast bacilli, red, and your blue background. So that's Ziehl-Neelsen staining. Okay, so heat is applied in that process. Okay, now apart from Ziehl-Neelsen and Kinyan's technique, we also have Pappenheim's, which makes use of rosolic acid in alcohol as the decolorizer instead of acid alcohol. So this technique differentiates Mycobacterium tuberculosis from Mycobacterium lacticala and Smegmatis. Smegmatis and lacticala are stained blue, while Mycobacterium tuberculosis are stained red. So if you notice, from the first technique up to the third technique, Mycobacterium tuberculosis are stained red. On the fourth technique, however, the Baumgarten's technique, which makes use of diluted alcoholic fuchsin as the primary stain, here, Mycobacterium tuberculosis are stained blue, whereas the other organism, Mycobacterium leprae, is the one that's stained red. Okay, so blue, Mycobacterium tuberculosis, red, Mycobacterium leprae. So that's for Baumgarten's technique. So the four methods for acid-fast staining, you have Ziehl-Neelsen, hot stain method. You have Kinyan, the cold stain method with wetting agent. Pappenheim's, which makes use of rosolic acid and alcohol as the colorizer, differentiates Mycobacterium from lacticala and Smegmatis. And Baumgarten's, which makes use of diluted alcoholic fuchsin as primary stain, differentiates Mycobacterium tuberculosis from Mycobacterium leprae. So here, Mycobacterium tuberculosis is stained blue instead of red. Okay, Mycobacterium leprae is the one that's stained red. Okay. Now moving on to culture method. So this is the step or the method of identification that follows microscopic technique. So in this method, we inoculate or we plant our organisms on artificial culture media to observe them, to observe their growth, colonial characteristics, to further identify the organisms. So this process would require the use of culture media. Okay, so culture media are useful when growing or propagating bacteria. Okay, so it usually provides the appropriate biochemical and biophysical environment. So the biochemical environment is actually dependent. No, the type of biochemical environment or nutritional environment that will be used in cultivating a bacterium would depend on its special needs, if there are any. Okay? Now there's a wide range of culture media available, okay, for us to use, depending on what organism needs to be cultured. Now, before we move further, allow me to discuss these two terms, which are widely used in the culture method. So when we say inoculum, so inoculum refers to the organisms or the microbes or the specimen that you're going to add or introduce into a culture media. Again, inoculum is the microorganisms or the sample or specimen that you're going to add to a culture media. So we call it inoculum. Okay, so those that you put at the tips of your wire needle or those that you collect using the wire loop is called the inoculum. A culture, on the other hand, refers to organisms or microbes that are growing in or on a culture media, like this. Okay, so this pink structures here is called a culture. Same as with this one, greenish structures that you see here is called a culture. So these are microorganisms growing on or in a culture media. Okay? A culture is divided into three types. We have pure, mixed, and stock. When you say pure culture, it refers to a group of organisms that is basically similar. So it's just one type of organism, so pure culture. Say, for example, for this one, this is a pure culture of Klebsiella, for example. Okay? And then this one is a pure culture of E. coli. Okay, so all the organisms on this plate is E. coli. All the organisms on this plate is Klebsiella. Okay, mixed culture would refer to a culture that's made up of different types of organisms. Okay, it's evident actually in the colonial morphology of colonial characteristics that they display when they are cultivated on media. Okay, so if you can see pink colonies and colorless colonies and white colonies, so these are technically made up of different organisms if they display different colonial characteristics. So that's mixed culture. Now, when we say stock culture, this is actually pure culture of known organisms or identified organisms that are kept in the laboratory for referencing purposes. Okay, so these are cultures of organisms that will be used as either negative or positive control. Okay. So colony, when we say colony, this refers to the macroscopic or visible masses of microbial growth that you see arising on the culture media. Okay, so they're usually arising from a single cell. So this one here is a colony, this one is a colony, this one here is a colony. The small one beside it is another colony. Okay, so technically there are three types of colony. We have mucoid colonies like this, so they appear slimy, watery, or glistening. Okay. We have smooth colonies with uniform texture. Okay, so this allows them to be easily emulsified with normal saline solution. And this one is obviously the rough colonies. Okay, so they're wrinkled, dry looking, okay, granulated. These are not easily emulsified with normal saline solution. Okay, so moving on to types of culture media. So definitely there are lots of culture media available in the market. And there would be a lot of different types of culture media that you will be using in the laboratory. So this can be categorized as to physical form or consistency, as to composition, function or purpose, and distribution. So we'll go through them one by one, but allow me to start with physical form. Okay, so according to physical form or consistency, we have liquid, semi-solid, and solid media. Now, liquid media are those that do not contain solidifying agents or agar. Okay, so these are technically water-based. They could be broth or milk or infusion. Okay, growth is positively identified if it appears cloudy. So this would be how it would look like without bacterial growth. So it's clear. Okay, but with bacterial growth, it appears cloudy. Okay, turbidity will be dispersed, meaning the organisms have spread out. Okay, and sometimes there could be particulate appearance. So some floating materials may be seen, okay, or seen within the solution or on the top. Okay, so that's those are the signs for positive growth on liquid media. For semi-solid media, it contains a small amount of solidifying agent, about 0.3 to 0.5% agar. Okay, this is best in detecting motility. Okay, so examples of this are those that are used for motility test. No? So we have motility test medium and the sulfide indole motility medium. Okay, so the SIM is used in a set of culture media for biochemical testing. Okay, so it detects not only motility, but it also detects indole production here, so indicated by a pink layer on the surface. Okay, and then hydrogen sulfide production is indicated by blackening. Okay, you'll know that the organism here is motile because the hydrogen sulfide has been is dispersed all throughout the culture media. Okay, if it's not motile, hydrogen sulfide might have been located only in a particular area, somewhere where you have inoculated the organism. So since this is semi-solid, we usually inoculate in a stabbing motion. So if it's not motile, the blackening can only be seen at the center, where the needle has touched the culture media. Okay. Moving on to solid media. So this one contains 1 to 5% agar. Okay, so it would allow the growth of discrete colonies. Okay, so a lot of culture media that will be mentioned earlier are used widely for growth of organisms. Okay, so we have liquefiable and non-liquefiable solid media. Now, when you say liquefiable, you can prepare it in advance in an Erlenmeyer flask. Okay, say a liter of Erlenmeyer flask, which contains the culture media. They can solidify. Okay, so if you're going to use it, you can just melt it up and then pour it in different Petri plates or Petri dishes. So this includes nutrient agar, McConkey, EMB, the usual line of media that are used or has been identified and mentioned previously. Now, non-liquefiable culture media, which includes Middlebrook 7H11 and Lowenstein Jensen media, both are used for the cultivation of Mycobacterium tuberculosis. Okay, so since Mycobacterium tuberculosis is quite fastidious, okay, these culture media contains nutrients, okay, and additives that are proteinaceous or protein in nature. So protein is quite sensitive to heating. Therefore, we do not reheat or remelt culture media such as this. So after preparing them, they should be plated at one time. Okay, so we cannot reheat them or remelt them again. So this will destroy the additives, okay, that contains which are high protein components, of this culture media. Okay. Then we have, according to composition, we have the synthetic and non-synthetic media. So culture media with compositions that are chemically defined are called synthetic. So a lot of the culture media that we use in the lab are synthetic. All those in bottles and powder, and then you have to reconstitute them. Those are synthetic media. Okay, so their components has been identified, therefore it's synthetic. Non-synthetic media, also known as complex media, okay, may have one or more component that are not chemically identified or is unknown. Okay, so it provides a full range of growth factors needed by an organism. Okay, so usually these are culture media that may contain blood or milk or yeast or beef extracts or heart blood components or heart meat components, no broth. Okay, so those are examples of non-synthetic media. Okay, but again, these are quite useful for cultivating pathogens as well as fastidious bacteria. Okay. Now, according to purpose, we have several types of culture media. Okay. So we have general purpose, like nutrient agar. We have enriched. When you say enriched, this is basically general purpose media with the addition of certain elements. Okay. So it's a solid media, enriched like blood agar plate and chocolate agar plate. Then we have enrichment. This is liquid media, okay, like trypticase soy broth and selenite F. Okay, selective media. Okay, so selective media includes EMB and McConkey. They contain components that limit the growth of gram-positive organisms and select only the growth of gram-negative organisms. Okay, they can also be differential as well. Okay, so EMB and McConkey can support the growth of gram-negative organisms and differentiate one from the other by virtue of their colonial characteristics. Okay, so later we will see what those are. Okay, so blood agar plates are also examples of differential media. Okay. Now there are also culture media that are used for cultivating anaerobes, okay, like Thayer Martin media. Okay. We also have transport media like Stewart's and Amy's. We also have culture media for assays like Mueller-Hinton agar and culture media for enumeration, which is widely used in food technology or in industrial microbiology, where they need to count the number of colonies. So example of this is the potato dextrose agar. So this is not widely used in routine bacteriology laboratory or clinical laboratory. Okay. So let's start with the general purpose media. So this is also known as basal media. Okay, so basal media supports the growth of a lot of organisms, most non-fastidious organisms. Okay, it is generally used for primary isolation of microorganisms. Okay, so technically it's designed to grow a broad spectrum of microbes. Okay, examples, we have nutrient agar and nutrient broth. Okay. Then we have enriched and enrichment. Again, the difference is that enriched is solid, while enrichment is liquid. So the addition of extra nutrients in the form of blood, serum, egg yolk, vitamins, and amino acids will make a basal medium enriched. Okay, that's why it's called enriched, because it's technically enriched with a lot of nutrients. Okay, so it supports the growth of numerous fastidious organisms. No? So examples of this would be blood agar plate, chocolate agar plate, and Thayer Martin. So we add blood. Okay, technically, the preparation for BAP and CAP is similar. It's just that when we add blood to blood agar base, no, or basal media, the basal media is slightly warm. It's not hot anymore. So the blood is intact. Okay. Whereas in chocolate agar plate, blood is added while the basal media is still hot. Okay, therefore, blood is cooked in the process, turning red into brown. Okay, so it appears like chocolate. So it's called chocolate agar plate, but actually you can't eat it. Okay, and then for enrichment, no, for enrichment media, this promotes the growth of a particular organism by providing it with essential nutrients. Sometimes it may contain certain inhibitory substances to prevent the growth of normal competitors like normal microbiota. Okay, so examples of this would be selenite F, brain heart infusion, and thioglycolate. Okay, then we have selective media. Okay, so selective media, okay, is designed to inhibit unwanted commensals or contaminating bacteria and help recover pathogens from a mixture of organisms. Okay? So selective media are technically agar-based. You know, any agar, for that matter, can actually be made selective by the addition of certain inhibitory agents that don't affect the pathogen of interest. Okay, so that's important. Okay. Various approaches to making a medium selective usually includes the addition of antibiotics, the dyes, chemicals, pH, no, alteration of pH, or a combination of any of these. Okay, so examples of which is mannitol salt agar. Okay, so mannitol salt agar is both a differential and selective media. It's selective primarily because of the presence of a high salt concentration, 7.5%. Sodium chloride is technically high. So it limits or inhibits rather the growth of those that cannot survive in the presence of high salt concentration. Okay. Hectone enteric agar contains bile salts that inhibits growth of most gram-positive organisms. Okay. So this is mannitol salt agar, and this is hectone enteric agar. Okay. So I have here a couple of tables that include some examples of selective media. So we have Mueller telluride. So it contains potassium telluride, which allows the growth of Corynebacterium diphtheriae and limits the growth of other organisms. Enterococcus faecalis has sodium azide tetrazolium. It's perfect for isolation of fecal enterococci. PEA, or phenylethanol agar, contains phenylethanol chloride and is widely used for the isolation of Staphylococcus and Streptococcus. Tomato juice agar contains tomato juice and acids for the isolation of lactobacilli. McConkey, of course, contains bile and an indicator, the crystal violet, which is perfect for isolation of gram-negative enteric organisms. Okay. SSA, or Salmonella Shigella agar, contains a variety of selective agents. No? Bile is among those. Okay? So it's useful for inhibiting growth of other organisms, but allows growth of Salmonella and Shigella. Okay. Then we have the Lowenstein-Jensen agar. It contains malachite green. And again, we use this for isolation of mycobacteria. Okay. Now, the last one is actually for fungal organisms. So Saborod's agar is selective because of the acidic pH. Okay, so 5.6. That's perfect for isolation of fungal elements. Okay, so moving on to differential culture media. Okay, so certain media are designed in such a way that different bacteria can be recognized on the basis of their colony color. Okay, so various approaches, including incorporation of dyes or metabolic substrates, are usually done. No? So those bacteria, okay, that can grow on differential media can utilize those additives and appear as differently colored colonies. Okay, so we call it differential or indicator media. Okay, because indicators are usually added, okay, to differentiate one organism from another. Okay, so differential media allow the growth of more than one microorganism of interest, but it can distinguish one organism from the other by virtue of the color of the colony, such as this one. Okay, so this is a McConkey agar. It contains neutral red as the indicator. Okay, so this allows a differentiation of lactose fermenters from non-lactose fermenting organisms. So those that produce pink colored colonies are lactose fermenters, and those that produce colorless colonies are non-lactose fermenters. Okay. Another example is the spirit blue agar, which contains a spirit blue dye and is widely used for identification of organisms that are able to utilize fats. So blood agar plate is also a differential media. So it differentiates the hemolytic reactions of some microorganisms. Okay? So we have here beta hemolysis, which is identified with the presence of a clear zone of hemolysis around the colonies. Okay? So clear, ideally, initially the culture media is red because of the blood that was added. Okay? But since the organism is able to make use of red blood cells, it's able to break it down, causing the clear zone of hemolysis. Okay? So beta hemolysis. And then on the next plate, you can see hemolytic reactions, because there's a change in color. However, there's a greenish discoloration that's visible. Okay? So if you look closely like this, okay, so it should be clear like this. However, okay, in this portion, there's a greenish area, okay, here indicating partial hemolysis of or partial destruction of RBCs. We call this alpha hemolysis. And then if there's no hemolytic reaction, okay, no zone of hemolysis, that's called gamma hemolysis. Okay, so alpha, beta, and gamma is differentiated in blood agar plates. Now I have here a table, a couple of tables showing additional examples of differential media like blood agar plate. So it has been discussed. So it differentiates between types of hemolysis. MSA differentiates species of Staphylococcus. So it contains mannitol. So it will be able to show staphylococcal species that are able to ferment mannitol and those that do not. Okay. HEA or hectonenteric agar contains bromthymol blue, okay, to differentiate growth of Salmonella and Shigella, as well as other lactose and non-lactose fermenting organisms. And then spirit blue dye, okay, contains spirit blue agar rather, contains spirit blue dye to differentiate fat utilizing bacteria from those that do not. Okay, and then urea broth, okay, so contains phenol red to differentiate those that are able to hydrolyze urea from those that are not. SIM contains thiosulfate and iron. It differentiates organisms that are able to produce hydrogen sulfide gas from those that do not. Okay, TSI or triple sugar iron agar contains triple sugars and the phenol red dye, which is the indicator to show or demonstrate sugar fermentation. Okay? And then iron, of course, is used to demonstrate formation of hydrogen sulfide. Okay? And then XLD here, or xylose lysine deoxycholate, allows differentiation of xylose fermentation, lysine decarboxylation, and hydrogen sulfide production. Okay. And then we have culture media for anaerobic growth. Okay? So anaerobic bacteria needs special culture media for growth because they need low oxygen content, they need reduced oxidation reduction potential, and of course, additional nutrients. Okay? So Media for anaerobes may have to be supplemented with nutrients like hemin and vitamin K. Okay? And such media may also have to be reduced by physical or chemical means. So we can boil them. Okay? It will, boiling, the medium serves to expel any dissolved oxygen. Okay? So before using the medium, it has to be boiled in a water bath to expel any dissolved oxygen. And then it is sealed with sterile liquid paraffin. Okay. Examples include anaerobic PEA, or anaerobic phenylethyl alcohol blood agar. It's an enriched selective media and widely used for inhibition of facultative gram-negative rods, and it also inhibits the swarming of certain Clostridium species and Proteus species from clinical specimen. Okay, and then we have the anaerobic kanamycin and vancomycin thioglycolate broth. So it contains the antibiotics kanamycin and vancomycin. Okay, and then we have Robertson's cooked meat, cooked meat media. Okay, so this one is commonly used to grow Clostridium species. Okay, it contains a bullock heart meat. Okay, bullock heart meat. A bullock is a castrated bull or young bull. Okay. So this is an example of an anaerobic chamber. So this is where we inoculate anaerobic culture media. So we just don't inoculate our anaerobic culture media anywhere. Okay? We need to observe the proper environment to allow our anaerobes to grow successfully in an artificial environment. So this one here is similar to a biologic safety cabinet. Okay? It's just that except for the glass, instead of the glass, there's a thick plastic sheet that covers these opening of the chamber. Okay? And then there are glove ports where you will insert your hand so that you can work on your culture media. So once you have finished inoculation, these plates will be placed in an anaerobic system. And then when you're done with that, you can now open the anaerobic chamber because the anaerobic system will have to be incubated. Okay. So these are examples of the anaerobic system. So this is gas pack. We actually have this in school. I don't know if they bought another one. Okay, so GAS PAK, gas pack, it's a brand name. So it contains palladium catalyst pellets. It contains envelopes that are supposed to be opened to produce carbon dioxide. Okay, so it has sodium bicarbonate and sodium borohydride, which would supply carbon dioxide. Indicator systems are also included so that we could check from time to time if the system is still anaerobic. Okay, so Petri dishes are placed in an inverted manner because moisture usually builds up on the cover. So if we have incubated it right side up with the cover on top and the agar-filled portion at the bottom, the tendency is that fluid buildup, moisture buildup will be found on the cover. So every time the system moves, okay, moisture will fall on the lower portion, which will contain the agar. So to avoid that, okay, to avoid moisture from dripping into your culture, drowning your organisms, we need to incubate your plates in an inverted manner. Even if you don't need to place them in anaerobic systems like this, plates have to be inverted every time you place them in incubators. Okay, if the organism has to be transferred, okay, you can place them in an anaerobic pack. So this contains a gas generator that will provide the anaerobic environment. Okay, so when you need to mail your samples, you can use this. If you don't have the gas pack, you can use a large jar that will allow Petri dishes to fit inside it. Okay, and then place or stick a candle on a glass light, light it up, close the jar. The candle inside will remain lighted so long as there's oxygen in the environment. And then when it has consumed the oxygen, the light will turn off. Okay, so that signifies that the system is already anaerobic. Okay. Then we have transport media. Okay, so clinical specimen may sometimes need to be transported to laboratories in other areas. Okay, so um, we need to make sure that the organisms we're going to transport are still viable. So we need to use or to plant our organisms in a culture media that will maintain its viability, okay, and would slow the rate of microbial reproduction. So we use transport media. Okay, so it prevents drying of the specimen and it inhibits overgrowth of unwanted bacteria. So a lot of transport media are actually nutrient poor. Okay, so it technically only contains what is needed to temporarily survive. So that when we use transport media, we have to be familiar with the limit, no time limit. Okay, so usually it's 2 hours. So within 2 hours from collection, the sample should be already in the laboratory. Otherwise, even if they're placed in transport media, they'd slowly die. Okay. So the examples of transport media are the following. So you have Stewart's. Okay? It was created 1954. So this transport media maintains original state of the specimen, especially if it contains Neisseria gonorrhoeae. So it prevents organisms from being exposed to heat, to cold, to drying. And then its swabs are also charcoal impregnated. It does contain glycerophosphate in the medium. Okay, so charcoal is added to neutralize inhibitory factors, but it may result to difficulty in gram staining interpretation. Okay, so again, we need to follow the 2 hour limit. That's also set for Stewart's because some gram negative bacteria may utilize the glycerophosphate in the medium and overgrow. Some of those organisms that we need to isolate might die in the process because remember, it only contains glycerophosphate. So if you take too long, longer than 2 hours, okay, the organisms of interest might slowly die off, okay, and will be replaced by overgrown microflora, okay, or microbiota. Okay. Then we have Amy's, 1965. This is similar to Stewart's. It's actually modified Stewart's medium. It has replaced glycerophosphate with balanced salt solution in the media. Okay. It retained charcoal, but instead of impregnating the swabs with charcoal, charcoal is added into the media. So it is believed to be a far better choice than Stewart's, for transportation. Okay, and then we have Carrie Blair. Okay, so Carrie Blair is also similar to Stewart's, but it's modified for fecal specimen. Okay, so the pH is also changed to 8.4 from 7.4. Okay, so the pH is increased. So this is perfect for fecal organisms. So charcoal is also removed in this setup. It's perfect for, or it is recommended for fecal specimen that is suspicious for Campylobacter species. And then we have buffered glycerol saline. Okay, so this is designed for stool samples only. Okay, it's widely used for mailing fecal specimen. Okay, it has high pH also and favors fecal pathogens. Okay, so fecal pathogens grow in highly alkaline environments. Okay, however, it's not recommended for transport of fecal specimen that are suspected to be positive for Campylobacter. Okay, so if you if you have suspicion that the samples might contain Campylobacter, it's better to use the Carrie Blair instead. Okay. And then for assays, so culture media for assays, okay, are usually used for assays of vitamins, amino acids, of course, antimicrobial agent. No, we use this to monitor the effects of administration of certain antimicrobial drugs. Okay, so example is the Mueller-Hinton agar. So this is the Mueller-Hinton agar. Usually it's placed on a larger Petri dish to allow zones of inhibition to spread out. Okay, so we use this for antimicrobial sensitivity testing. Okay, and then we have for enumeration. Again, we don't use this in routine clinical laboratories, but this is widely used in industrial laboratories, no, for food technology. Okay, so they use this for testing, no, food samples for the presence of microorganisms. So they need to count the number of colonies on it. Okay, so example of culture media for this purpose is the potato dextrose agar. Okay, so additional information. Okay, so for enumeration, okay, there are some techniques and instruments that are needed to perform enumeration, okay, or counting of uh, microbial organisms on the culture media. So this would include direct microscopic counts. Okay? So these are possible using special slides known as counting chambers, similar to what we use in hematology, the Neubauer chamber. Okay? Then we have the electronic counting chambers. So it counts numbers and measure size distribution of cells. But this is more widely used in hematology. Okay, and then, of course, we have indirect viable cell counts, which are also known as plate counts. So it involves plating out or spreading out of a sample of a culture on a nutrient agar surface. Okay, and then the sample or cell suspension can be diluted in water or saline before plating. Okay? So if plated on a suitable medium, each viable unit grows and forms a colony. And each colony that can be counted is called a colony forming unit or CFU. Okay? And the number of CFUs is related to the viable number of bacteria in the sample. So this is what we did in urine. No? So in specimen during specimen collection, in the discussion for specimen collection, pour plate is actually used. Okay, and then the number of visible colonies were counted. Okay, so in the same manner, you can also count with the use of grids, like this, and then we can use the colony counters or culture counters. Okay. Then moving on, we have media according to distribution. Okay, so culture media according to distribution, we have tubed and plated. So those are placed on Petri dishes are called plated media. Okay, and those that are placed on tubed are called tubed media. So tubed media may be full butt, okay, or we call this agar deep. Okay, or they can be plain slant, no butt. Okay, so plain slant. So this is used widely for transportation of organisms or for stock cultures, keeping stock cultures. You can place them here or grow the organisms here. And then we have the slant and butt, okay, tube media. The slant and butt tube media is widely used in biochemical testing, like this one. This is TSI agar. Okay, so this is used for biochemical test. Okay, now to continue, now we have methods of obtaining pure culture. So this is our goal when we culture. So we wanted to isolate pure culture of organisms to be able to identify them properly. So how can we do that? How can we obtain pure cultures? Now the easiest way, widely used way, is the strict plate method. Okay, so it's best for isolation of pure colonies on solid media. Okay, so these are pure colonies that you see here. Okay. Then you can use the pour plate method. Okay, so pour plate method has also been discussed, no, similarly, no, in specimen collection when we discussed urine. Okay, so placed 1 ml of the sample in a sterile empty petri dish. Okay, and then add the slightly warm culture media, swirl them together to mix them properly, incubate them, and then observe for growth the next day. Of course, since you were able to mix the specimen together with the agar, so growth will be observed not only on the surface, but there are also subsurface colonies. Okay? Now, apart from the pour plate method, we can also use selective method. So simply using selective media. Okay, so you will be able to isolate a particular group of organisms by using selective media. Animal inoculation is also used. However, we don't perform this in routine clinical laboratories. Again, animal inoculation is widely performed in our reference laboratories or in research laboratories. Okay. So how do we inoculate? No, how do we inoculate pure cultures or cultures on culture media? So how do we plant our organisms on culture media? So it depends on the type of media based on distribution. So if you have tube media, okay, and you have liquid or broth, okay, oh sorry, broth, inoculation is done by mixing a loopful of the microbe or the solution that contains the organism into the liquid media. Okay. Semi-solid, like SIM, okay, is inoculated through stabbing. So we make use of wire needles instead of loops. So we stab. We touch the wire needle to a colony, and then with that attached to at the tip of the needle, we stab it directly onto a semi-solid media. Now for solid tube media, it has both butt and slant. So you can stab and you can streak. Okay, so stabbing and streaking. Remember when you stab, do not touch or do not push the needle up to the bottom of the tube. Okay, provide an allowance. So do not streak, do not stab completely up to the bottom. Now for plated media, okay, we can inoculate our organisms in different ways. Okay, so through streaking. Okay, so the manner of inoculating on plated media is streaking. So we have quadrant streaking. This is the common way of inoculating on plated media. The quadrant, because we divide the surface of the media in quadrants. Okay, so we have 1, 2, 3, 4 quadrants. Okay, and then you can also use this. Okay, so continue streak. You can start in this area and then move towards the center, stop at the center. Okay, start again on the other side and then move towards the center. Or you can use gradient streakings. In MH agar or Mueller-Hinton, we use this to perform sensitivity testing. So when we do so, we plant, we inoculate on the entire surface area to produce a map of bacterial growth. So our target is to allow the growth of bacteria to spread all throughout the entire surface area of the Mueller-Hinton agar, so that we inoculate on the surface using overlapping radiant streaks. Okay, so making sure that all the surface is covered with microbial organism. So again, as mentioned earlier, the most common way of inoculating on plated media is the quadrant streak. Okay, so in quadrant streaking, you only touch your loop in the sample once. So you'll only collect inoculum once. Okay, so to start off, make sure to practice aseptic techniques. So sterilize your wire loop. Once it's sterilized, okay, gather your inoculum and streak on the first quadrant. After streaking, heat. Okay, you do not need to gather another loop full of organisms. So you'll be getting your organism from the first streak. So after heating or after sterilizing your loop, allow it to cool a little and then allow it to touch on the shoulders of the first streaking. Okay, allow the loop to touch here and drag the streakings towards the next quadrant, just like this. Okay, and then heat, sterilize your loop. Okay, after, allow it to cool for a while and then continue with the third quadrant. So for the third quadrant, allow it to touch a portion of the strict second quadrant and then drag it towards the third quadrant. Okay. Then hit again to sterilize the loop. Allow it to cool for a while and then allow your loop to touch, okay, the shoulder of the third quadrant streaking, and then allow the streaks to spread out for the last quadrant. Now, this is because we want to isolate colonies. Okay, so after inoculation, you incubate. The next day, check for colonies. So this is how the colonies would spread out, with this type of streaking method. So this is an actual culture media and an observed. Okay, so this is the first quadrant streak. Look how thick and thick the colonies are and close together. And then this is the second quadrant streak. The third quadrant streak is not as close together. You can actually see some separate colonies. And then this is the fourth quadrant. And then this is the fifth quadrant. Okay, so you can actually see separate colonies because on the last quadrant, you'll be streaking a little more loosely to allow them to spread out, not as close as this one. Okay. So that's how we streak on plated media. Okay, so finally, preserving bacterial cultures. If you need to preserve your bacterial cultures, you can deep freeze them at -50 to -95 degrees Celsius and lyophilize them, by freezing them first, -45 degrees Celsius to -72 degrees Celsius, and then vacuum dehydrate them. Okay. So before we end, let's recall what we have learned for today. So you were introduced into the different methods of identification, which would start with microscopy and staining, followed by culture, then biochemical test, serological test if needed, and then we now have molecular testing. And for research laboratories, you have animal inoculation. So for this discussion, we were able to cover microscopy and culture. So microscopy is done with the use of staining, of course, for enhanced visualization. Okay, so we have direct staining and indirect staining. Direct staining includes simple, special staining, and differential staining. But among the three, differential staining method is more commonly used. Okay, so because this includes gram staining and acid-fast staining. Okay, so indirect method includes negative staining, where the background is stained and the organisms or the object to be viewed is left colorless. Okay, so types of media. So you're also able to discuss the types of culture media. Okay, so culture media may be classified according to physical form. So we have liquid, semi-solid, solid. According to distribution, we have tube and plated. According to composition, we have synthetic or chemically defined. And then we have those that are not chemically defined, non-synthetic. Okay, so non-synthetic media. Okay, and then we have those that are based on functional types. So we have the general media, general use media, or the basal media. We have enriched, enrichment, selective, differential, assay, enumeration, and we also have transport media. Okay, and then of course to isolate pure culture. So this is the goal of culture cultivation, or the culture method. Isolating pure cultures is done by performing strict plate method, poor plate method, selective media, and animal inoculation. So inoculation in a strict plate method can be done, in quadrant streaking. You can also do continuous streaking, or you can do the lawn technique, especially for assays like sensitivity testing. Okay, so what else? You're also able to tackle um, inoculation on tube media. So for semi-solid, we simply stab them. For liquid rather, for liquid media, a loopful, we mix a loopful of the inoculum. For semi-solid, we stab them. For solid media, on solid tube media, we can stab and streak our culture media. Okay, and that ends our discussion for part 1, methods of identification. Okay, so stay tuned for the part 2 of methods of identification that will cover the rest of the methodologies that were previously mentioned. So again, thank you for listening, and God bless.